welcome everyone to OHBM 2022 and to the educational course on generative modeling of brain dynamics from principles to application. I'm Adeel Razi, and I'm the co-organizer of this educational course with Daniel and Marinazzo. We have 8 lecturers, and each lecture will be about 40 to 45 minutes. We will have about 10 minutes after every lecture for Q&A, and then we will wrap it up at the end. The main learning objective of this educational course is to teach the attendees the basics of the generative modeling, its basic principles, practices, recommendations, and implementation, and also the applications of some of the representative generative modeling techniques. So I will start with the first one, and the first lecture will be about dynamic causal modeling. By the end of this first lecture, you should be able to place dynamic causal modeling in the analysis pipeline. You should be able to tell the differences between different connectivities, for example, the structural, functional, and effective connectivity. You will be able to explain how a generative model helps to separate the bold signal into its constituents, for example, the neural activity, hemodynamics, and the noise. You should be able to explain the interpretation of different parameters in the neural formula that I will be driving for DCM for fMRI, and explain the different parameters and log model evidence can be used to test model hypotheses. Okay, so let's start. So that's the agenda for today. I will start with the overview of DCM. Then I will talk about neural models and hemodynamic model, and here I will drive from the first principles the neural model used in DCM for functional MRI. I will then talk a bit more about model inversion and second level and the second level analysis, and then I will finish by quickly showing you or pointing you towards a worked example that's in the SPM manual. Okay, so let's start with the DCM overview. So DCM is a Bayesian framework, and it is used for inferring effective connectivity. And in the brain. So this is how do we define DCM. Importantly, we talk about the neural responses in the brain because they are in this lecture, because there are certain applications of DCM which are outside, for example, for modeling COVID-19, and there is some recent work on modeling climate change even. So here we will only, of course, focus on modeling effective connectivity in the brain. So where the DCM sits in the pipeline. So this is how do we start. So we start with an MRI machine where we acquire our structural and functional brain images. We then use these brain images and then do image processing, which involves various different processes, for example, realignment, registration, normalization, smoothing processes. We then, once we have done all of the pre-processing, we then put this into a GLM to calculate certain contrasts, so to look at different differences between different experimental conditions. Once we know where the activation is happening, we can extract the time series from those specific brain regions, and then we can use these time series to put them into a model and look at and go from activity to connectivity. Yeah, so here we can now set up a model of how different neural populations will be interacting with each other, and this is where the DCM sits. So how we can define various different sorts of connectivity. So here we have 3 different sorts of them. So first one is the structural connectivity. And by definition, this is basically showing the structural connection between brain regions. So here in these cartoons, I'm using 3 brain regions, and structural connectivity can be thought of as presence of axonal connection, and it can be inferred from using diffusion imaging, for example, using probabilistic or deterministic spectrography techniques. This is structural connectivity. In this lecture, we will be focusing more on functional MRI. So one of the most widely used connectivity type is functional connectivity. And which we define as statistical dependencies between regional time series. So here we have 3 brain regions or 3 brain time series, and we look at the correlation between them essentially. And by definition, these correlations are undirected. Yeah. And then the third part, or the third type of connectivity, is the effective connectivity. Here we look at the direction of the information flow, yeah, and also the balance of the connectivity, like is it excitatory or inhibitory. So this is basically defined as the causal influences between different neural populations, and by definition, it can only be inferred. Yeah, you cannot measure it, but you can only infer it for the reasons that I will explain on the next slide. So let's say, what is the system of interest? So the system of interest in many of the in all experimental designs with functional MRI, you have some external stimulus. Yeah, for example, you have a visual stimulus or a auditory stimulus, which, and here in this example, we're showing people a person who is lying in the scanner, a screen which shows lots of dots. Yeah, you can model these external stimulus with a deterministic vector U. For example, here is showing is the stimulus is there or not, or off. Yeah, this external stimulus enters the brain, for example, and it changes the neural activity. Yeah, so you see this or you hear this stimulus, and it changes the neural activity. Yeah. However, this neural activity is unknown. So we cannot directly measure this. This is inside the brain, right? So we can't directly measure this. We can only infer this. Yeah. But what happens is that this neural activity passes to the hemodynamics, it changes the blood metabolism, and then it is picked up by our scanner as a functional MRI or the bold signal. And what we see is this vector Y, which looks like what was given as an input, but it's of course slightly modified or distorted while it went through various different stages and before it was recorded by the MRI scanner. So our main task is to actually try to infer from what we have measured and what went in to guess what actually would have happened at the neural level. And this is what we can only, as I said, infer. We only measure this, and we try to infer what actually happened at neural level. And this means that we can, because we have to infer this, which means that we have to have a model or a generative model to be specific here. The generative model that we use in DCM is a hierarchical one. So there is a neural model, as I showed before. There are different brain regions, and we are interested in how they interact. Yeah, this is what's happening at neural level. So this is how do we model this? How brain activity X, some activity in brain region 1, let's say X1 or X2 or X3 or X4, how it changes over time. So as the time passes, as the person lies in the scanner, time passes, we want to see how this activity is changing over time. And mathematically, we show this as a differential equation or a difference equation in discrete time. So X dot is equal to, is a function. So X dot is the change in the activity over time, which is the function which depends on what's happening now. So X is the current activity, U is the input, what experimental input has gone in, and U and then theta is the connectivity, how different elements of this model are interacting with each other, and this theta represents the effective connectivity. Now this what's happening in the neural at the neural level, it passes through the hemodynamics, and we will talk about the observation model, and then it gets recorded by the scanner. So this is basically what we see. The Y is our observed signal. And G is our observation model or the hemodynamic model, which is dependent on the current activity and the hemodynamic parameters. Yeah. So this is, and together these two models, the neural model and the observation model, they give you what we call a generative model. Yeah. Sorry. So the generative model is basically defined by two things: the data, yeah, and what the input is. So this is how we define our generative model. Now, in practice, what you have is your observations. Yeah, so you observe Y, the bold signal, and then you want to infer these model parameters. Yeah, what is the effective connectivity? What were the different parameters of this hemodynamic model? And you, as I said, you have to have this generative model, that how do we go from the external stimulus to your observations. Once you have established this generative model or the forward model, what you do is this, that you take the observations and then you do a process, what we call a process of model inversion. And basically what you're doing is this, that given your observations Y, or the bold signal, and your experimental input U, you try to infer that what parameters theta would make this model fit to the observed to the observed data. And this is as I said, this is the model fitting procedures or model inversion, and the specific algorithm we use to do the model fitting is called variational expectation maximization algorithm, or variational Bayes. So in the end, what we want is this, that DCM is a hypothesis testing tool. So for example, if you have one model where you say that, okay, this is the model, there are 4 different brain regions and they are connected in this way, and there's an external input which is shown by a red arrow here, it enters here, and then we have another model. So this is model 1, and then model 2, where we say, okay, these 2 brain regions have an extra connectivity here, and we want to compare these two models and want to find out that which model, either the model 1 or the model 2, explain our observed data. So this is basically what the DCM allows you to do, which to do a model comparison. Yeah. So a quick intelligent summary. So what do we do is this, that so each of the model we can thought of as a hypothesis. So you embody your hypothesis in a generative model. Yeah. And this generative model allows you to separate the neural activity from hemodynamics. Then you perform an inversion. So you fit each model to the observed data. When you fit this model, you get the model parameters and also something what we call the model evidence, or the and then you use this model evidence to make decision on which model is better model. Yeah. And then you can look at the model parameters, and then you can make your interpretation. So that's in a sense, in a nutshell, what the DCM framework allows you to do, which is to specify different hypotheses, test those by fitting them to the model, and then allowing you to do a Bayesian model comparison to choose between which model is the better model that explains your model the best. Okay, so now we go into the specifics of what sort of neural models that are in use in DCM. Okay, so that's as I previously showed, this is what we call a system of equation that's describing the neural activity over time. Yeah, so X here is neural activity ongoing. 1 to n, n is a number of brain regions, and then we say that this change in activity in a certain brain region is a function of current activity X, the input U, and the model parameters theta. There are various different sort of DCMs in use. So I will be basically focusing on deterministic DCM for fMRI, or the task DCM, and I will be driving this formula in the next few slides. This is in a specific form of DCM where we use a bilinear approximation. Yeah. Then there is I'll talk a bit briefly about DCM for CST, or resting state fMRI, or the spectral DCM. This is again a very popular flavor of DCM. We'll briefly touch on this. And then there is a new canonical microcircuit-based DCM for fMRI, which uses multimodal multimodal data from fMRI and also EEG or MEG. Okay, so let's start and talk about driving these neural formula for the deterministic DCM, or the bilinear DCM formula. So let's start. So we start with a certain specific experiment. So what we're interested in, that how does the brain activity X change over time? So that's the main question that we are asking. Yeah. So let's say that we have a certain experimental paradigm where we have some visual stimulus, some dots, moving dots on the screen. Subjects view some moving dots using during fMRI. So on some trials, the subjects were instructed to pay attention to the speed of the dots motion. So these dots are moving, and during some of the trials, we asked participants to pay attention to the speed of the of those dots which are moving. Now, what we're interested in, what is our research question, is how does this attention to motion change the strength of connectivity between various different brain regions, for example, V1, V5, and superior parietal cortex. So this is basically we start to put together different elements of our model. So here we have 3 brain regions. We say, okay, there's a V1, V5, and SPC, and we identified them by using a first level usual GLM analysis. Once we have these brain regions and their time series, we start to put together our generative model. So let's start here with a very simple one region DCM. So let's say we have a region, the primary visual cortex V1. There's a driving input U1, so that's the visual stimulus which is entering, and X1 is the activity in this brain region V1. Yeah. So if we want to say how we can write down the change in activity in X1 over time, then we can simply write it down. So first, let's say we look at this U1. We are showing that there are this is our external stimulus looks like. It turns on for a brief time and then it goes off. Yeah. The X1 would look like something like this. So there is so once there is an input here, it goes up and then it slowly goes down. It decays, and then it goes up and then it decays. Now, if I would want to show this, then we start to put some parameters of a model. So we say that the strength of the connectivity of this driving input is we defined by C, and there's a self connection here, or self connection we defined by A here. So these two parameters are the parameters of our model, A and C. And if we want to now write down the changes in brain activity X1, then we can write it down with this simple formula. So change in activity X1 dot. Yeah, so this is differential equation, and this is equal to A times X1. So because this A is the self connection, so the change in X1 is a function of how much the self connectivity A is, plus what is the external influence. So there is some external stimulus U1, and then there is a coefficient C here, which shows the strength, how much of this actually goes into the region. Yeah. So this is if, for example, A is large negative number, so it means that there would be very, very faster decay. Yeah. So, and if A is not as big, then this decay would be not very fast. It would be very slow decay. Yeah. And if it's if A is positive, then there would be no decay, but rather it will go up, which implausible. This self-activity works like a dampening, and always tries to, or a negative feedback, and always tries to dampen what's happening in the brain region. So this self-connectivity is always modeled as an inhibitory influence. Okay, so this is very simple. We had only one brain region. Now, what we do is we make it a bit more complex, and we add another brain region like V5. Yeah. Now you could start to write down the equation. So if you want to say what's the change in x1, so we already have an equation for this. Yeah. Which is the self connection A11 times the what's happening in the brain region x1 plus some coefficient C. Yeah. An external coefficient, external input, and then it goes into the brain region, x1 or V1 here, and that's how we write down the first one, as we saw in the previous slide. And then we want to see what's happening. What is the change in activity in region 2? Yeah, which is the V5. And now you can, the same way that you wrote that this equation, you can now write down this equation as well. So this would be self-connectivity A22 times X2 plus, now you don't have an external input directly entering V5, but you have what was happening in X1, it's now entering into X2. Yeah, so you write it down in a similar way that you wrote this, but now this would be A21. So this is the connection going from V1 to V5, and we write it down as A21 and times X1. So this is the first part, is the self-decay, and then V1 input. Yeah, so this is how do we write to these two equations, each describing the change in activity in those brain regions. Now, we love matrices, right? So we now put this thing into a matrix form. So we just concatenate these two activities, and then we write it down in this form where the columns are outgoing connections and the rows are incoming connections. Yeah. So this is how we define the structure of this model, and you can then write this more concisely as x dot equal to A, which is now a matrix, and x plus C, which is again now a column matrix, and then you have the external input U1. Now, okay, so we can now write the system of equation in this form. Yeah, but let's make it a bit more complicated. Let's say you remember, okay, so before we make it a bit more complicated, let's just so remember there was a U1 like this, then there was an X1 like this, and then we could expect that what's happening in X2, this X1 goes in, and then it further, it still remains, it has similar, similar shape, but slightly different now. Is more, there's a more smoothness in this. Yeah. So this is how X2 would look like. Now let's make it a bit more complicated. And now remember what we had in this experiment is this, that there's an attention to motion component. Yeah. So we now have an external, another external factor U2, which is an attention. And now this is, we say, okay, what this attention is doing is modulating the connectivity between V1 and V5, which is this connection A21. Yeah. So if we are to now think of this, then there are two inputs, U1 and U2. U1 is a same input as before, and U2 is an attention. So for some trials, this attention is there, for others it's not there. So if X1 was like this, now you could expect that the connectivity, the activity in X2, which was previously some smoothed form of this X1, now it's actually modulated by the attention. So you could see that it's slightly down here, but then it goes up as the attention goes up, and then it comes back. Yeah. So can we now model this interacting what's happening here in the form of equation as we did before? So let's try to do this now. We could again write down the change of activity in V1. So we can still write it down as x dot x1 dot. Sorry, there's no x1 here. This should be x1 dot equal to a11x plus c11 u1. However, if we want to write down what's happening in V5, now it has an additional component. So remember, there was self-connection, there was some contribution which is coming from x1, and then there's another contribution which is coming due to the attention. Yeah, due to U2, which is an attention a stimulus, and it's modulating this connection, which is to this connection. And we can write it down as, let's say this coefficient is B21 because this is acting on this connection. And then you have U2, which is the attentional thingy. And then you have this this activity which is traveling from x, which is entering in x2, and this is x1. So this we can now write down as self-decay. There's a V1 input and there's a modulatory input. So there are three components now to the which we use to model the change in activity in x2. So if we want to now write down this system of equations, then for, let's say there were m inputs, so we say let's say there are many there were more than one modulatory inputs, then we can just simply write it down as a summation of all those different modulatory impacts, and then we have something like this. So we have an external stimulus U, and we write down the overall system of equation as this bilinear form. And this is what the usual DCM formula looks like. You can write it down as this. So if you want to say how the A looks like for this specific model, it would have these parameters A11, A21, A22. Then we have a one modulatory input, U2, and then we have one external input U1, and that's how we write down this formula here. This side is capturing the change in activity per region. Then you have this A matrix which shows the structure of this model. Then you have this modulatory input and the modulatory connectivity parameters which are in this connectivity matrix B. You have activity per region, X1 and X2. You have a driving input C, and you have all external stimulus or external input. So this is how we have now derived this Taylor approximation, or the bilinear form of DCM, which is widely used. Okay, so now once we have this neural activity, remember that we now have a neural formula. Now we have to add to this what we call an observation model. Yeah, so remember this neural formula, we just drive this form specifically for a task-based DCM for fMRI, and now we have the second component is this observation on the hemodynamic model. So this is what we call a balloon model. So what it is basically saying is this, that you have this neural activity. It changes it, and it changes it engenders the vasodilatory signal, and then this would in turn change the flow, the blood flow, and this blood flow then changes the blood volume and also changes the oxyhemoglobin content of the blood, and this then subtends the Bolt signal. So this is what we call the hemodynamic model. The hemodynamic model and the neural model that we drive together, they give you the generative model. Yeah. So this so now we have derived this more formula. Now let's quickly look at resting state DCM. So in resting state DCM, for example, there's a one specific flavor of DCM called stochastic DCM, which is very similar to what we have as you see in a deterministic DCM. Only this now that we now also have added a component which we call WX, and WX are the random fluctuations, or they actually account for the endogenous fluctuations or the thoughts. So if there is a resting state where you don't have an explicit U, for example, you could add this and these neural fluctuation as a proxy for the thoughts, and then these are the driving force that changes the activity in X. Yeah. In this situation, we can use this stochastic DCM for fitting resting state data, but usually this requires solving this equation. This is no more a simple or what we call an ordinary differential equation, but because of this this uncertainty added by the neural noise, this is what we call a stochastic differential equation. And these system of equations is usually very hard to solve. It becomes very slow. So this specific form or flavor of DCM has been in literature for a while, but because it's slow, although it's very powerful, and I don't want to go into much details of this, but it's a very powerful tool, but it's just less used. Yeah. So there is another flavor of DCM for resting. This is what we call a spectral DCM, and this is basically more widely used. So if we look at this stochastic differential equation that I was showing you before, and for a specific case of resting state DCM, it means that we don't have an external input U, so you can put U equal to 0. So if this U is 0, then this term goes on, this term goes out, and what we are remain with is this simplified form. Yeah, where we have X naught equal to AX plus some neural fluctuations. This equation, or this system of equation, is slightly simplified. What we did is that instead of solving the system of equation in time domain, we use a trick, and where we take all of this into a Fourier domain or a spectral domain, and we solve this system of equation in frequency domain. So that's a trick we used to speed up the inversion process, the computational efficiency of the process, which results in graphs which are we can invert or estimate graphs which are larger. Yeah, we have inverted as big as graphs with containing 30 to 40 nodes. There's another version called regression DCM introduced by Klausino Stefan and Stephen Frazel and colleagues, which can even actually invert models as big as hundreds or thousands of nodes. So this is a specific form of DCM used for resting state fMRI. Then once you have done a DCM fitting at the subject level, then you want to do an inference at the group level. And then I will just quickly show you a bit of this as well. As I said, DCM is a Bayesian framework. So in a Bayesian framework, you have 3 different quantities of interest. So you have some prior. So this is a prior knowledge that you have about your system that you know. This is where you start. These are your prior beliefs, and you put them into a P of theta. So this is what we call prior, and here we show this as a green curve. Yeah. Then what you want are your posteriors. So you want to know, if I have priors, what are my once I see some data, which we define here with a generative model, probably of a probability of y given some model parameters theta. This is called likelihood, and this this embodies the generative model. Once you have seen some data and you have prior, you want to see what is the posterior. Yeah, probability of observing your model parameters given some data y. Yeah, so these are the three constituents of any Bayesian model. These are the posterior estimates that we are usually interested in. Yeah, so these are the effective connectivity. When you do a model fitting or the model inversion process, the estimation of a model gives you, as I said, the posterior probability. So these are, for example, for every connectivity parameter, you have a posterior probability distribution. We use Gaussian priors, and we make use of some of the properties of those of these statistical framework, where we have Gaussian priors and we have Gaussian posteriors as well. This is called a Laplace approximation. So you get these parameters where they are not point estimates, but actually distributions. What you also get after model inversion is some form of a score of which shows how good your model fitting is. Yeah, and this is what we call model evidence, probability of y given some model m. And usually this model evidence, we can't calculate this exactly, but we rather calculate a lower bound on this evidence. This is what we call the free energy, and free energy can be defined as a trade-off between model accuracy and complexity. Yeah, so these are the two things that you get. You get model parameters, they're posteriors, and also you get model evidence, and then you can use this model evidence to do Bayesian model comparison. Yeah. So if we are interested in group level inference, then what we do is this, that we fit DCM for each subject, yeah, in your data set. Yeah, and this is where we have these DCMs. Yeah, what we do is this, we create this first level model. Yeah, and here we also have measurement noise. So this is a GLM at the first level. What do we do is this, then we take these first level DCMs into a second level GLM, or a second level model, in which we have at the second level, again, this is a GLM, where this X is your design matrix, where I will talk a bit more about this on the next few slides. This shows you, this defines your what your design is. And then here we have again an error term, which basically where we try to model the between subject variance. Yeah, so this is a hierarchical model, first level DCMs, and then we go to the group level, and then at the group level, at this level, we have some we also define some priors on these parameters theta, which are the group level effects that we are interested in, and this whole framework is called parametric empirical Bayes. Yeah. So this is a hierarchical model that we use for group level inference in DCM. So quickly, so this is the GLM that I was showing on the previous slide. And this is how this design matrix X look like. So you have, for example, you have many subjects, and you put your and this is where you concatenate all the subjects data here. You define your design matrix here. These are your model parameters that at the group level that you are interested in, and here you have between subject variance that you have these error terms here. And so this is basically a Bayesian GLM that you define. Here you have group averages for each connection. Then you have, for example, you are interested, you have let's say 3 groups, and some of these columns will be modeling the group difference between the group 1 and group 2. Then there could be you are also interested in the differences between group 1 and group 3, let's say. So there could be another, or there could be furthermore here, depending on the question that you have. So basically, what we're doing at the second level is to estimate these parameters theta. So these are your group level effects that you are after. And once you do this analyses at the group level, you get this screen which describes various different things that I was talking about on the previous slides. It tells you your design matrix here, how many covariates you have included. For example, here there are 5 covariates. How many DCM parameters were there in your model? There are 12 of them, for example, in this one, and how many subjects you included in the analysis. It shows you the group level parameters. So this is what we are interested in, and you can choose what level of threshold you want, and so on and so forth. So this is basically a summary of second level analysis. So if I quickly summarize, DCM is a framework which enables us to make inference about the effective connectivity of brain regions, which and the important point is this, that the effective connectivity can't be directly observed. We can only infer this using a generative model. Yeah. Usually what we do is this, that we embody each of our hypotheses into a generative model, which we can then invert and then compare them. Yeah. So this is and this takes us to what we call the Bayesian model comparison, or the Bayesian model selection. So this is the last part of our talk, where there's a worked example in the SPM manual. I don't have time to go through showing you practically how it's done, but if you go to the SPM website, download the manual, depending on which version you have downloaded, it would be either 36 or 30 chapter number 36 or 38, something like that, which gives you a detailed instructions on how you can do first level analysis, where you can define, for example, here 4 nodes of a default mode network, MPFC, PCC, and bilateral inferior parietal cortices, and then do an analysis using a data set that we have also provided. So if you go to the chapter, there's a link to this data which you can download, and then you can follow the chapter guidelines. You can go step by step, and it will help you to actually implement this first level DCM for a default mode network. Okay, so as I said in the beginning, the learning outcomes of this lecture are that you can now place the DCM in the way it is in the analysis pipeline. You can talk about hopefully you can understand different 4 sorts of connectivities which are in literature. Then you know now how to set up a generative model, how to invert that, how to do the interpretations, and so on. So with that, I would like to thank lots of people, especially the methods group at UCL, and lots of this, almost all the slides were created by the members of the methods group, for which I'm thankful. My lab at Monash Computational Neuroscience Laboratory, and also Carl Fiston. Okay, thank you. Bye-bye.